after being done with glycolysis in the Krebs cycle, we're left with 10 NADHs, 10 NADHs, and 2 FADH2s. And I told you that these are going to be used in the electron transport chain, and they're all sitting in the matrix of our mitochondria. And I said they're going to be used in the electron transport chain in order to actually generate ATP. So that's what I'm going to focus on in this video: the electron transport chain. And just so you know, a lot of this stuff is known, but some of the details are actually current areas of research. People have models and they're trying to substantiate the models, but things are happening at such a small scale here that people can just look at the evidence, some of it of which is indirect, and say this is probably what's happening. Most of this is very well established, but some of the exact mechanisms of, for example, how exactly some of the proteins work aren't completely understood. So I think it's very important for you to understand that this is kind of at the cutting edge. You're already there. So the basic idea here is that the NADHs, and that's where I'll focus, FADH2 is kind of the same idea, although its electrons are at a slightly lower energy state, so they won't produce quite as many ATPs. Each NADH, maybe I'll write it here, each NADH is going to be, as you'll see, indirectly responsible for the production of 3 ATPs, and each FADH2 in a very efficient cell, in both of these cases, will be indirectly responsible for the production of 2 ATPs. And the reason why this guy produces fewer ATPs is because the electrons that he has going into the electron transport chain are at a slightly lower energy level than the ones from NADH. So in general, I just said indirectly. How does this whole business work? Well, in general, NADH, when it gets oxidized, so NADH, remember, oxidation is the losing of electrons or the losing of hydrogens that happen to have electrons. We can write its half reaction like this. Its oxidation reaction like this. You'll have some NAD+, which you can then go and use back in the Krebs cycle and in glycolysis. You have some NAD+, you'll have a proton, right? A positive hydrogen ion is just a proton. And then you'll have 2 electrons. This is the oxidation of NADH. Oxidation of NADH. It's losing these 2 electrons. Oxidation is losing electrons. Oil rig. Oxidation is losing electrons, or you can imagine it's losing hydrogens from which it can hog electrons. Either one of those is the case. Now, this is really the first step of the electron transport chain. These electrons are transported out of the NADH. Now, the last step of the electron transport chain is you have 2 electrons, and you can view it as the same 2 electrons if you like. 2 electrons plus 2 hydrogen protons. And obviously, if you just add these 2 together, you're just going to have 2 hydrogen atoms, which is just a proton and electron, plus 1 oxygen atom. So I could say 1/2 of molecular oxygen. That's the same thing as saying 1 oxygen atom. And you're going to produce, if I have 1 oxygen and 2 complete hydrogens, I'm left with water. I'm left with water. And you could view this, we're adding electrons, or we're gaining electrons to oxygen. Oil rig, reduction is gaining electrons. So this is the reduction of oxygen to water. This is the oxidation of NADH to NAD+. Now, these electrons that are popping out of, these electrons right here, that are popping out of this NADH, and when they're in NADH, they're in a very high energy state. And what happens over the course of the electron transport chain is that these electrons get transported to a series of, I guess you could call them transition molecules, but these transition molecules, as the electrons go from one to the other, they go into slightly lower energy states. And I won't even go into the details of these molecules. One is coenzyme Q, and cytochrome C, and then they eventually end up right here, and they're used to reduce your oxygen into water. Now, every time an electron goes from a higher energy state to a lower energy state, and that's what it's doing over the course of this electron transport chain, it's releasing energy. It's releasing energy. So energy is released when you go from a higher state to a lower state. When these electrons were in NADH, they were in a higher state than they are when they bond to coenzyme Q. So they release energy. Then they go to cytochrome C, release energy. Now that energy is used to pump protons across the cristae, across the inner membrane of the mitochondria. This is all very complicated sounding, and this is the cutting edge. This is the cutting edge, so it maybe should sound a little complicated. So let me draw a mitochondria. So let me draw a small mitochondria just so you know where we're operating. That's its outer membrane, and then its inner membrane, or its crista, would look like that. Maybe it looks just like that. And let me zoom in on the membrane. So let's say if I were to zoom in right there. So if I were to zoom that out, that box would look like this. You have your crista here, and I'm going to draw it thick, right? So I'm zooming in this green line right here. I'm going to draw it really thick. I'm going to color it in with green, just like that. And then you have your outer membrane. This outer membrane, I can do it up here, and I'll just color it in. You don't have to see the outside of the outer membrane. Right here, this space right here, this is the outer compartment. And then we learned in the last video, this space right here is the matrix. This is the matrix. This is where our Krebs cycle occurred, and where a lot of our NADH, or really all of our NADH, is sitting. So what happens is every time NADH gets oxidized to NAD+, or and the electrons keep transferring from one molecule to another, it's occurring in these big protein complexes. And I'm not going to go into the details on this. So each of these protein complexes span. So let's say that's the protein complex where this first oxidation reaction is occurring and releasing energy. And then let's say there's another protein complex here where the second oxidation reaction is occurring and releasing energy. And these proteins are able to use that energy to essentially pump, it might all seem very complicated, to essentially pump hydrogens into the outer membrane. It actually pumps hydrogen protons. Let me be very clear. Hydrogen protons into the outer membrane. And every one of these reactions pump out a certain number of hydrogen protons. So by the end of the electron transport chain, or if we just followed one set of electrons, by the time that they've gone from their high energy state in NADH to their lower energy state in water, by the time they've done that, they've supplied the energy to these protein complexes that span our cristae to pump hydrogen from the matrix into the outer membrane. So really the only byproduct of the oxidation of NADH into eventually water, or the oxidation of NADH and the reduction of oxygen into water, isn't ATPs yet. It's just this gradient where we have a lot higher hydrogen proton concentration in the outer compartment than we do in the matrix. Or you could say that the outer compartment becomes a lot more acidic. Remember, acidity is just hydrogen proton concentration, the concentration of hydrogen protons. So the byproduct of this, all of this energy is used to really just pump these protons into the outer membrane. So you have two things. The outer membrane becomes more acidic. than the matrix inside. Maybe we could call that basic. And obviously these are all positively charged particles, so there's actually an electric gradient, or electric potential between the outer membrane and the inner membrane. This becomes slightly negative, that becomes slightly positive. These guys wouldn't naturally do this on their own. If this is already acidic and it's already positive, left to its own devices, these more protons wouldn't be entering. And the energy to do that is supplied by electrons going from high energy state in NADH to going to a lower energy state eventually on the oxygen in the water. That's what's happening. But essentially all that's happening is protons being pumped from the matrix into the outer compartment. Now, once that gradient forms, these guys want to get back in. These guys want to get back in to the matrix, and that is where the ATPs are formed. So there's a protein that also spans this. Let me draw. So remember, this is all this inner membrane right here. Let me just draw it a little bit bigger right here. So that's our inner membrane, our cristae right there. There's a special protein called, and I'll show you actually a better diagram of what it looks like in a second, called ATP synthase. ATP synthase. And what happens is, remember, because of the electron transport chain, we have all of these hydrogen ions up here. All of these protons, really, all they are is a proton, that really want to get back into the matrix down here. They really want to get back down to the matrix, but they can't. This cristae is impermeable to them, so they have to find a special way to get through. They were able to go the reverse direction through these special protein complexes. Now they're going to go back into the matrix through ATP synthase. So they're going to go back, but something interesting happens. And this is really an area of current research where people are, they think they know how it works, but they're not sure, because you can't just take these proteins apart and operate them or watch them operate like you can a regular mechanical engine. These are ultra small, and they have to be in a living system, and they have to have the right conditions, and you can't even, it's hard to see hydrogen protons. These are ultra small things that are, pretty much you can't see them. But what happens, the current model is as these enter, as these go through my ATP synthase, there's actually an axle. So you can kind of view this as a housing, and then there's an axle, and this is all just a big protein. There's an axle, and then there's another part of the synthase down here. So you can imagine, this is kind of mind-blowing that something this fancy is occurring on the membranes of pretty much all living systems' cells. It's not just eukaryotes, even prokaryotes do this. They don't do it in mitochondria, they do it in their cellular membrane, but it's a pretty neat thing. And what happens is, as these go through, you can kind of imagine as water flowing through a turbine, it mechanically causes this structure in the middle to spin, to actually spin. This is the current thinking. And this thing is all uneven. It's not like this nice tube, it'll look all crazy like that. And what happens is that an ADP molecule, let's say that this is the A part of the ADP, and then you have two phosphate groups, it'll attach to one part of this protein, and maybe a phosphate will just randomly attach to another part of this protein, just like that. So right now it's just ADP and a phosphate. But as this inner axle turns, because it's not a symmetrical tube, it has different things sticking out that has different amounts of atomic charge. It's going to play with this outer housing right here. And so as this turns, the outer housing, because of just the proteins bumping against each other and electrical charge and whatever else, it's going to squeeze the ADP and the phosphate together to actually form ATP. And actually, the current thinking is it does it on 3 different sites simultaneously. So as it spins around, ADP and phosphate groups kind of show up on the on the inside of this housing. You can imagine like that. And I don't even know if it's on the inside, but they show up on the housing. And then as this thing spins around, it stretches and pulls on this outer part and pushes these two things together. So it's using the energy from this proton gradient to drive this axle. And because it's all strange, it does all these distortions on this outer part and actually pushes the two ATPs together. So when you start off with your 10 NADHs, it'll provide just enough energy to do just enough protons to put into the outer membrane that when they go back through our ATP synthase, you can almost do our ATP synthase motor, just based on people's observations, they see that this will produce on a per NADH level roughly 3 ATPs. On a per FADH2 level, roughly 2 ATPs. And I've said multiple times in the videos, this is kind of an ideal. But a lot of times, maybe you'll have some protons leak, so their energy can't be captured properly. Or maybe some of these electrons might somehow jump the gun or jump some steps, so some of the energy gets lost. So you don't always have a completely efficient system. And just so you believe that this is actually occurring on our membrane, there's actual visual depictions of these proteins. This is the actual protein structure of ATP synthase right here. That is actually ATP synthase. And as you can see, there's this piece right here that holds this part, that holds that part, and that part. You can kind of imagine it relatively stationary. The hydrogen comes through here, the axle gets spun, and as the axle gets spun, ADP and phosphate groups that are lodged inside this F1 part of the protein get pushed together. You have to put energy into the reaction in order to make them stick together, but they get pushed together by the protein itself as this axle turns around. And this axle turns around from the energy of the hydrogen going. I don't even know what the mechanics would look like, but you can imagine, in my head, I imagine kind of, the simplest thing is a windmill, or not a windmill, is maybe some type of water turbine, or maybe the simplest thing is, if you have something like that, I don't know if that's what the protein actually looks like. You have any kind of thing passing by, it's going to spin it. It's going to spin it like that, and you could be more creative if you want to change the angle of the spin and whatnot. And that's all. People are really still trying to understand this at a deeper and deeper level. But for your purposes, especially at an introductory biology level, you just have to realize that 2 things are happening in the electron transport chain. Electrons are moving from the NADHs and the FADH2s to eventually show up and reduce the oxygen. And as they do that, they're releasing energy. As they go from one molecule to another, they're going to lower energy states. That energy is used to pump hydrogen protons into the outer compartment of the mitochondria. And then that gradient, those hydrogen protons want to get back into the matrix of the mitochondria. So as they go back in, that drives this ATP synthase engine, which actually produces the ATP. So just like we said in the past, when you have 10 of these, on average, let me say it this way, on average, each NADH is going to produce 3 ATPs, not directly. It produces enough of a gradient of hydrogen protons to produce 3 ATPs in the ATP synthase, and each FADH2, on average, produces enough of a hydrogen gradient to produce 2 ATPs. So if we come in with 10 NADH, they're going to produce, in this ideal world, 30 ATP, and then our 2 FADHs, H2s, are going to produce 4 ATP. And then if you remember, from glycolysis, we had 2 net ATPs directly produced, and from the Krebs cycle, we had 2 ATPs directly produced. So then you have 4 from glycolysis and Krebs. And that gets us once again to our magic 38 ATP from one molecule of glucose. And now I think you have a pretty good grasp of cellular respiration.